This is the Cell Cycle by me.
And me. Before we get in, this is a cell we are using. It is very simple because art is very hard. Since it is 2n equals 6, it has 3 unique chromosomes for even more simplicity.
The interphase is split into 3 parts, G1, S, and G2. The interphase is the longest part of the cell cycle, if it ever makes it there. G0 phase, a period of rest between the cell, either temporary or permanent, and it can't divide in the state. Here are some cells that can't do mitosis and are mostly in G0 phase. But for the case of our cell, it is an interphase.
Currently, the cell is in the G1 phase, which you might think stands for growth 1 phase, but it's not. It's called the gap phase even though it's growing. It produces organelles and proteins for later use.
Then, the cell gets into S phase. The S phase stands for synthesis, replicating DNA for the other cell.
The cell now contains 2 times more information. It is still 2n equals 6 because there are no unique chromosomes being created. I hope. The single chromosome now has a duplicate attached to it using a centromere. And since there is still 6 centromeres, it still has 2n equals 6.
The final phase, G2, is the last checkpoint before the cell breaks apart. It also duplicates its organelles that aren't related to the DNA, such as the mitochondria. If it fails here, it does apoptosis.
The cell doesn't actually explode. The parts of the cell membrane break off and the neighboring cells clean up its mess.
If we didn't do apoptosis and actually passed the checkpoint, we are now ready to split apart.
Now that it has everything to go into M phase, it will. There are 2 steps, mitosis and cytokinesis. This is how the cell divides. Prophase, metaphase, anaphase, and telophase. You can remember these phases by remembering it or PMAT.
Prophase is the first part of mitosis and the longest. 1/2 of the total time of mitosis is used here, just like you with your homework. Except the cell finishes and you don't. All it is the removal of the nuclear envelope. And it condenses the duplicated chromatins into sister chromatids, which are attached by a centromere. While that is happening, meiotic spindles from the centrioles, made out of microtubules, form from the centrosomes at the poles which pull the chromosomes apart later. Later as in after metaphase, later.
Metaphase is where the chromosomes line up across the middle of the cell. Think of the M in the word as middle to help you remember. Come on guys, this isn't supposed to be like the CSW lunch line. The centrosomes are connected to spindle fibers which come out of the centrioles that help with their positioning.
This is a very important part because it makes sure the daughter cells receive the correct amount of genetic material needed. Misalignment of chromosomes in this phase can lead to genetic disorders for the person because of the uneven split.
Next is anaphase. The sister chromatids are now moving away from each other. This is the split that gives the daughter cells their chromosomes. Now the cell has 12 chromosomes, double the amount because you count it by the centromeres. The spindle fibers are disassembled by the end of anaphase.
Finally, the last stage of the cell cycle, telophase and cytokinesis. Now, if you're wondering how that fits to PMAT, telophase and cytokinesis happens at the same time. The spindle fibers are gone, and the chromosomes become less condensed, and a new nuclear membrane forms around both of the chromosomal areas.
Cytokinesis is when it separates into 2. Cleavage furrow happens in animal cells and helps them to divide. It is a groove on the cell where a contractile ring made of actin filaments will form. This ring will pinch the cell's plasma membrane inward, which will eventually split the cells entirely.
That is how one cell becomes 2 through mitosis. So, for recap, if you got all that, very impressive.
If you didn't get all that, you should rewatch the video.
If you want to know more, ask Dr. Valenti. He knows more about this stuff.